Sure. Okay, very well. I mean, I oh, is it right? Okay. I did.
Oh,
Planner, yes.
just a baby. No, so since 2013. It's not up to me. I know it's uncomfortable.
So they're wanting to do well, my visa expires on October 1st.
Welcome, everyone. It's so great that everyone could make it today. We're here for Suyen, who's presenting her thesis today, and then after this hour, doing a private defense with her committee. So Suyen worked on biofilms. She did a really amazing job working on that. She created a huge data set. She developed a new way to study biofilms. Streptococcus pneumoniae, one of the major causes of respiratory infections, otitis media, which is ear infection, and she really dedicated everything to that. And Suyen also arranged for herself actually to be funded throughout the years by all these amazing private foundations, which was really great and which also provided her a lot of freedom in the lab. And with that freedom, she also decided to be involved in Boston Bacterial Meeting, one of my favorite conferences in Boston, maybe the best conference in Boston. And at one point, she actually made it to be the boss of the Boston Bacterial Meeting. So she could direct everybody around, which was great because she also did that in the lab. Boss everybody around. Nah. Well, that's what everybody always told me. So.
You don't have proof of that.
So she did a great job, and she's going to tell you all about it, right? So take it away. Good luck. You'll do amazing.
Thank you everyone for coming today. So as Tim said, I've been working for the past 6 years on trying to study bacterial biofilms, and you'll see by the end of the talk why I got fascinated in studying biofilms. And bacteria in nature, they actually grow as communities and as cell aggregates. And I always like to picture this and imagine these bacterial cells, these what we call biofilms, these cell aggregates, they mirror image just like a human city, where each individual cell in the city has their own function, their own job, their own profession, that at the end, at some point, they will help in the community. So now, but how are these biofilms growing? It does start with planktonic cells. This is how we usually grow cells in the lab. We grow them as liquid media. And mostly everything that we know about bacteria, we know them from these planktonic cells experiments. So these planktonic cells will find a surface and they will attach. Once they attach, they will start producing this, what we call a matrix. And this matrix is helping bacteria to stick together, but also it protects them from the environment. Once this biofilm continues to grow, at some point, we don't know exactly when or how, but some bacteria will detach from the biofilm and will release what we call dispersal cells, and these dispersal cells are the ones in charge to keep the cycle going and keep the biofilm maintaining. So what is happening in the context, so once planktonic bacteria, I told you that they attach, but once they attach, they interact with the surface. As they interact with the surface, they're going to be generating phenotypic variants, and these variants are going to be locking down within the community. As this is happening, also, they're going to be interacting with the cell, with their neighbors. So imagine, of course, you live in a city, you have to interact at some point with your neighbor, you have to say hello. Same bacteria, they have to interact with their neighbor, and as they interact, it creates a suitable environment for horizontal gene transfer, creating a lot of genetic traits. So now I just told you that they are phenotypically and genotypically diverse. So some of the causes I mentioned, cell-to-cell interaction, and of course the structure of the biofilm will generate heterogeneity. And as this heterogeneity within the structure also creates a nutrient gradient, meaning that these cells that are at the bottom of the biofilm, they are unable to get the same amount of nutrients that the cells that are at the top. So now these are the causes, but what will be some of the consequences of this complexity? And for the purpose of my talk, I'm going to focus on two things, two consequences that are related to pathogenic bacteria. The first one is the interplay that biofilms have with antibiotic tolerance and resistance. So it is well known that in general, biofilms are more tolerant and more resistant to antibiotics. And this is due to this matrix, this layer that protects the bacteria that is embedded within the biofilms. And then, of course, because of this matrix, they're also good at evading the immune system. And as Tim mentioned, I focus most of my work on Streptococcus pneumoniae, and strep colonizes the human respiratory tract. It's still, even though we have developed multiple pneumococcal vaccines, still a problem, causing a huge burden in medical costs. But also, in addition to that, if we add the problem of antibiotic resistance, there are over 1 million resistant infections caused by Streptococcus pneumoniae per year, just in the US alone reported. Of course, this bacteria causes biofilms in the human body, and it causes biofilms in the nose, the heart, the lungs, and also the ear, really telling us that, of course, it's extremely important to study these bacteria in the context of a biofilm, since they are part of the infection cycle. Now, the issue with studying Streptococcus pneumoniae biofilms is that most of the field have focused on early in vitro assays. So really, we don't have a lot of knowledge on those genes that are involved in the later stages of formation and maintenance. So this is where I started, and I started thinking about how can we have a platform or something that will allow us to grow these biofilms for longer. If you think about it, when you have an infection, you just don't have it for a few hours. Your infection can even become chronic, and then you will need further treatment. So today I'm going to tell you 4 different stories, and the first one is the key one and the one that allowed me to do the rest of them. And I'm going to tell you how we developed in the lab a long-term in vitro biofilm assay that now these platforms allowed us to do a full characterization, not just in vitro, but also in vivo, and looking at the transcription of these bacteria under biofilm conditions. Then, because now we're able to grow biofilms for longer than just the attachment phase, then we did a time-lapse course experiment to actually try to identify what were those genes required for biofilm formation. So if you think about it, bacteria has a lot of genes. Are there any specific set of genes that are required for making a biofilm? Yes or no? And what these genes are. And now, because we're able to keep growing this for longer, I took a look at how these biofilms were growing for over 40 days, and in addition, to try to understand how they were developing resistance to antibiotics. So I'm going to start with the first part of the story. And after multiple trials, we changed. I'm not going to tell you the many things that we changed, but what I'm going to tell you today is what we came up with. And I told you before that in order to form a biofilm, we need to start with planktonic cells. So indeed, that's how we start. We have our planktonic bacteria. And the approach that we take is that we use a 24 well plate. In each one of these wells, there's going to be a glass cover slip. Basically, what is happening is that planktonic bacteria is attaching to these cover slips, and then it will keep maturing and form a mature biofilm. It is important to say that in order to keep this growing and maintaining, we have to come in the lab every single day and babysit them and change the media. Okay, but we can keep doing this for so long. And after day 4, we do what we call it's a passage, where we take the old biofilm and we pair it in a 6-well plate with a new clean cover slip, in this case the pink. And basically what will be happening is that those cells that are detaching from the old biofilm will find a new surface to colonize in the pink cover slip, and they will, this is the way that we can keep promoting biofilm growth. And we can keep growing this virtually forever. So here I'm just showing you an example of how biofilms, so we can take a look at these biofilms under the microscope, how biofilms look like after each one of the passage. Okay, now, great, we're able to grow biofilms. What else? So I got intrigued into, can we actually take a look at the different ways that they're growing? How are they growing? And to do that, we use confocal microscopy. So thank you, Brett, for all the help there. We stain our biofilm. So anywhere you see color, that means that the bacteria is stained. And we use 4 parameters to look at our biofilms. The first one is biomass. That's going to tell us how much bacteria density is attached in the cover slip. Then we look at average thickness, and that's just going to let us know about distribution of how many cells are in each one of the layer of the biofilms. Then we look at maximum thickness, so that's it's gonna be measuring from the bottom to the top peak of the biofilm. And then roughness coefficient, which is basically a metric of how flat the biofilm is growing. So for example, this, or how uneven or rough the biofilm is. So a rough biofilm will be characterized by these peak and valleys. So the first focus was on two strains that were previously reported as high and low biofilm. While the method that they used was not fully quantitative, so we decided to put these two strains. So here again, whenever you see green, it means that bacteria is there, it's attached. So this is how it looks in our assay, the high biofilm former BS72, and the low one, you see little green there, so there's not many bacteria attached. In our assay, when we quantify biomass, I'm just showing you here one of the four metrics, we can actually confirm that the BS71 is indeed a low biofilm, and it's showed by the lower biomass numbers. And now imagine we have four different parameters. In order to make this reproducible, we use we take multiple pictures, and so we have this over the course of 4 days. So how do we actually try to make sense of the data and try to reduce the data in a way that we can visualize it? And to do that, we perform a principal component analysis, and this is helping us to say two things. First, that the replicates, they group together. And second, that we are able to separate that high from low biofilm formers. And then, after taking a look at those two different strains, I went into the lab and I started working with my two favorite strains. And the first one is Tiger 4. Tiger 4 is a bacteremia model, meaning that the bacteria will quickly migrate to the blood. And then BHA97, which is an otitis media model, meaning that the bacteria has been used for ear infection models. So again, I grew biofilms, I quantified them, and the first thing that we noticed is that Tiger 4 forms smaller biofilms compared to BHA97. And now if we plot this data, again, into our principal component analysis, we were able to actually separate also these other two different strains, really telling us that this approach that we're taking it's separating different strains with different biofilm formation capacities. Oh. It's not. So to conclude this part of the story, I don't know, okay. To conclude this part of the story, I just show you that we developed a platform, an assay that was allowing us to grow biofilms for longer than just the usual 24 hours. And then I also show you that we use 4 different parameters to quantify these biofilms. In addition, our approach of looking at all these data sets together was able to identify strain-specific biofilm formation capacities. Now, I want to take you back to how biofilms are growing. And remember that they start with planktonic cells, and this is how we usually look at them in the lab, just in liquid media. And I just show you how these different bacteria are growing as surface attached, as biofilms. But then remember, there's a third component, and that third component are dispersal cells. So I got curious to really understanding what these dispersal cells were growing and what was the role in within the biofilm. And it is worth to mention that these dispersal cells, if not much is known about the biofilms, there is even less known about dispersal cells. So I want to take you back again to my assay. And basically here, I'm just doing a zoom in into one of the wells. So I'm showing you here how biofilms are attached to the cover slip, and basically dispersal cells will be those floating around. So I took these cells out, I stained them, and I looked at them under the microscope, and what we saw was that these cells are in a mixture of aggregates and single cells. So you can actually see here not much at day 1, but towards day 4, there are these huge chunk of bacteria there. So really telling us that these bacteria are detaching not just as planktonic, they're not single cells, they are a mixture of both. So really telling us that this is a new cell state. And then I came to wonder, and I want to take you back to the 2 consequences, remember, the antibiotic tolerance and resistance story and the immune evasion. So I then wonder, okay, they look different, planktonic cells, biofilm, and dispersal cells are different, they look different. I wonder what was the antibiotic sensitivity of these 3 different cell states. And to do that, I focused on antibiotic tolerance. And first, I want to say antibiotic tolerance is not antibiotic resistant. An antibiotic resistant bacteria can grow in the presence of antibiotic. A tolerant bacteria cannot grow. And that means that the minimum inhibitory concentration, so that concentration of antibiotic that is needed to kill the bacteria, it's the same for a susceptible and a tolerant strain. But how can we measure tolerance? We can measure tolerance by doing what we call a killing assay. And what we're looking at in a killing assay, it's really that difference on the amount of killing time that will take for the antibiotic to get rid of 99% of the population. So, for instance, a susceptible bacteria will have a lower minimum duration of killing, and a tolerant bacteria will have a higher duration of killing. So, taking that into consideration, I went back to my assay and I came up with this. I grew biofilms. I then separated biofilms from dispersal cells. I added antibiotic, and then I calculated the survival fraction of the final over the initial number of cells at multiple time points. And of course, thanks to this, we were able to make our killing assay curve. And here, first, I'm going to direct you to this purple line, which is the planktonic. And for the planktonic, you can see that the duration of killing, it's really in between 6 and 7 hours. But when we take a look at the biofilm population, this duration of killing increases. So it takes longer for the antibiotic to kill 99% of the biofilm population. Really, the dramatic change, it's focused on the dispersal ones. The dispersal cells, it takes almost double the time that it takes for the planktonic population to die. So really telling us that both biofilm and dispersal cells are less sensitive to antibiotic treatment. And we did this for multiple antibiotics. I don't have time to show you today, but it is true regardless the different type of antibiotics. So now I show you about how they grow. They look different. They have different antibiotic sensitivity profiles. What happens in vivo? So the next question was, do these different cell states, planktonic, biofilm, and dispersal, have a different disease outcome in an in vivo model? So for this, we take our 3 different types of inocula, in this case planktonic, and 3 days old biofilms and dispersal cells. We then infected a group of mice, and at the end we sacrificed them and we took samples from 3 different tissues, the nasopharynx, the lungs, and the blood. In addition, we were able to measure the severity of disease by using what we call a clinical score. And just to direct you here, a clinical score of 0 will mean that the mouse is a healthy mouse, it's not showing any symptoms, and a clinical score of 7 will mean a lethargic mouse. And the first thing that we notice here is that mice that were inoculated with dispersal cells, look at the clinical score here, so remember that the higher, the sicker they are. Those inoculated with dispersal cells show severe symptoms as early as 14 hours post-infection, really telling us that all these bacteria that are coming from dispersal cells are in a ready-to-cause disease state. But now if we go further into the actual bacterial cell loads in each one of the three different, either the nasopharynx, lungs, or blood, we can actually see that the three different inocula, they were able to colonize the nasopharynx. But when we take a look at lungs, biofilms are unable to migrate to the lungs and even to the blood. What we can see is that for the lungs, these dispersal cells showed a hypervirulent phenotype, really telling us that not only they're quick at causing disease, but they're also quick at making it to the lung. So now I show you how they look different, they have different antibiotic sensitivity profiles, and they also cause disease. So in order to try to understand what was exactly happening between these three different states, we chose to look at the transcriptome of these bacteria under the three different conditions to see what was happening. And the first thing that we did is, again, using our same sample or the inocula type that we use for our in vivo studies, so planktonic and 3 days old biofilms and dispersal cells. We extracted RNA and we prepared for Illumina sequencing, and then the type of data that we get from here, we get to compare our biofilm and dispersal populations, again, planktonic, generating what we call a differentially expressed genes. So for each gene, they're going to be experiencing, if they are experiencing a change, we were able to calculate that. And the first thing that we notice is that, okay, now here, each dot, it's a gene. If the gene is colored in pink, that means that it's differentially expressed, so it's experiencing some changes.